gene is a genetically modified organism, and it contains a gene from an entirely different species after a method which is known as gene transplantation, or different kinds of methods are there. So basic, what do you call it, transgenesis means transferring DNA into the animal or altering the DNA of the animal. So a transgenic animal is a genetically modified animal or organism that contains a gene from a different species following gene transplantation, or that results from the molecular manipulation of endogenous genomic DNA. So let us have a brief history or outlook of it. Before molecular genetics has been, come into place, they have tried to isolate the genes. Of course, there is a history wherein we started with isolation of genes, isolation of DNA, discovery of it, then how the double helical model of DNA was discovered. Then after that, a lot of biotechnological applications took place. Then from, you can say, from the 2000 onwards, in fact, there's a lot of change when it comes to genesis, but it was during the years of around after 1960s, that is during 1970s, that the first chimeric mice were produced. So from then onwards, people tried to do a lot of gene, try to understand the function of every gene. How is it, if at all we stop the function of a gene, that is knocking out its function, they are known as your knockout genes or knockout mice, in which such kind of genes were, whose function was stopped. Did it have an impact? Or suppose we incorporate a gene, and what is the kind of impact on the progeny or in that mouse? All this was they had been trying to see. So first transgenic animals were mice. They were obtained by micro-injecting genes into nuclei of one-day-old embryos. It was Ralph Brinster and Richard Parmiter who had first or identified or produced the first transgenic mouse with a phenotype. They were one Ralph Brinster was from the University of Pennsylvania, and Richard Parmiter was from the University of Washington. So what did they do? They tried to construct or incorporate a gene that was having this rat growth hormone, and this was under the control of a zinc-induced metallothionein promoter. So that what will happen with the inclusion of this particular gene, you could see that because of the inclusion of growth hormone in this particular gene along with your metallothionein promoter, there was a phenomenal growth in the physical structure, that is the phenotype of the rat. What did they exactly do? You can see on the screen, there's this rat growth hormone gene, and then you have a flanking sequence apart from that, or the right next to it, and along with that you have the metallothionein promoter. So this promoter ensured that this rat growth hormone gene got expressed. So it was injected into a fertilized mouse embryo, and this resulted in a transgenic offspring. And this, when fed with extra zinc, this turned out on the metallothionein promoter and resulted in the expression of the growth hormone gene, and because of which the rat grew twice its normal size. So you can just see that this was and it was published in Nature in the year 1982, in fact. So this giant mice instilled a lot of excitement in the scientific community because this was the first transgenic mouse. So Ralph Brinster and Richard Parmiter were the ones who developed the methods to transfer the foreign genes into the nuclei or the germline of the animals. And they had written a paper, Dramatic Growth of Mice that developed from eggs micro-injected with metallothionein growth hormone fusion genes. So the role of a promoter is to see that the gene was expressed. And it is still widely used today to develop different kinds of genes, different kinds of phenotypic results that are necessary, say enhanced insulin production, enhanced growth, and all such kind of, there are so many things that are taking place right now. And this has been extrapolated, that is extended to other mammals also from the year 1985. So right now you have transgenic mammals, especially rats, pig, sheep, and now you have fish. So Danio radio is one zebra fish, wherein what they did was they included translucent or luminescent hormone in it, so that it started glowing in the dark. So they are known as glowfish. We'll just come to that. And now you have a transgenic mouse which is glowing in the dark also. Thanks to this, today we have different kinds of models which are see, if you induce a hormone or induce a gene that is going to cause or suppress the tumor, or that is going to enhance the tumor, because of which even the mouse model is going to be affected, because of which you can actually study the disease also. At the same time, any kind of you can induce certain kind of drugs to be expressed, so that the immune system in the mouse itself starts acting upon it and try to kill the tumor that has been that is there in the mouse. Anyway, when you see the timeline, you can just see first transgenic livestock were produced in the year 1985, and then it could work. It was around 1989 that you could see that transgenic pigs express the human growth hormone, and they kind of suffered health problems also. So you have a side effect, because this is not something which is entirely normal. You are inserting a gene into a totally foreign a genome, because of which what is going to happen is that animal which is going to express this gene might have certain side effects, no doubt about it. So finally, pharma industry also started looking into this. This is known as genetic engineering, in fact. And then by 1996, Dolly was created. And then you have different kinds of livestock species, which were which started emulating the, or which became the models for human diseases also. And then you have the first transgenic livestock that were produced using the nuclear transfer technology. And 2000, you had gene targeting in the livestock, and transgenic livestock produced around, what are the different kinds of environmental, or what are the different kinds of concerns that are going to be seen. That is there. And then knockout mice were produced. And by 2003, you had the first gene knockout homozygote in the livestock. So, brief history of it. Now, let us just see. Often referred to, what is a transgenic animal or why do we need it? So that you can characterize the ability of the promoter, so that you have that particular gene to be expressed for sure. So you have a reporter gene as well that is present, and a promoter gene. So see here, LAC-Z is from your lacopron, and GFP is your green fluorescent protein. These are the promoters. They are attached to the reporter gene, so that whenever there is a green fluorescent protein attached, you for sure know that there is fluorescence going to be there. So you know that the gene has been expressed. And then you will start looking at whether how much of expression is taking place. Is there any kind of under or over expression or misexpression, in fact? Because in case when two genes are the combine two genes give rise to one product, then the entire the genetics are going to change. Is that gene expressing, or is there any kind of side effect with that gene expressing along with the gene next to it? Have we inserted it in the right place? All this takes into has to be taken into consideration. So misexpression of endogenous or foreign genes at specific time, location in the animal is also important. So finally, when we look into it, what are we doing? Why do we want transgenic animals in the first place? We are looking at the increase of the production of different animals in a short amount of time. And that is your to increase the production efficiency. Second thing is molecular farming, wherein you use livestock so that medicines, nutraceuticals, that is enhanced. Say, for example, you want vitamin D or vitamin A enhanced milk. So if you feed vitamin A, is it going to be coming in the form of milk? And then vitamin A enriched milk altogether, instead of you directly adding vitamin A to the milk. And then what about the tissues for transplant? Like I told you, they could create a complete ear for the from the different types of cells, so that it could be used for xenotransplant, that is transplantation. So all this is taken into consideration. Now, what are the different kind of strategies to produce this? Of course, you have two things, which is either you are going to give it a function to take place, or you can stop that function. For example, P53 gene is knocked out, then there is every chance for the P53, since it is locked out, it is basically used in or it is damaged in cancer. It is one of the genes that can be mutated or that can be stopped, because of which, if its function is altered, there is a chance for cancer to occur. So in case you stop the function or expression of P53 gene, then will it enhance the production of cancer or suppress the production of cancer? That has to be seen. Give me a moment, please. Yes. So when I stop the function of P53 gene, what is going to happen is it is a tumor suppressor gene, because of which, if at all I stop its function, or it's known as loss of function, then tumors will occur in the body. So in this case, if at all I give another apoptotic genes gain of function, suppose it is not being expressed in normal cell bodies. For example, podocytes or nephrotic cells in the nephron are there, which are not being expressed in the case of normal cells. In fact, in the case of a normal healthy individual, but they are expressed when a person is diabetic. So suppose I give it a gain of function in the normal healthy people, then they surely will get diabetes. So that is how, with the loss and gain of function strategy, whatever that is the result you are looking for, you'll be seen. And then at the same time, if you give growth hormone supplements in the form of, or you see that this gain of function in the growth hormone gene after adulthood is there, then overexpression of the gene product is there. So basic idea is that you add a cloned fragment of the DNA into the animal's genome, because of which the product over here, the growth hormone, that did not actually exist earlier. So if you give gain of function in a dwarf, specific to growth hormone, you see that the dwarf might be able to attain a certain amount of height. So that is there. And another paper also, the same paper has been published regarding it. And then when you come to silencing or loss of gene function, like I told you, this is going to be gene disruption. That is because of homologous recombination between the host genome and exogenous DNA. Now, when it comes to the different kinds of methods of gene transfer, you see that pronuclear injection is there, transposons, viral technique, that is retroviral or lentiviral, then sperm-mediated embryonic stem cells, chemicals, electropore. So you have basically physical, chemical, and mechanical methods of gene transfer. Different methods you can just see. So first is, you can just have a look at it. This is the DNA microinjection, in fact. So into the embryo and the pronucleus, you're inserting the DNA. Transposons and lentiviral vector with the help of either different the genes. Then ICSI is one more method, and then we will come to it in some time. And then you have a nuclear transfer. So with that, we will just have a look at it now. So DNA microinjection, what is happening? 1,000 copies of the foreign gene is taken in 1 to 2 plasmids, and then it is going to be injected into the pronuclei of 1-day-old mammal embryos. So this is going to have a first thing is superovulation of the females, and then this is going to be with mating by male, because of which whenever there is superovulation taking place, many egg cells are produced at the same time, which is not going to be seen in a normal case. Usually any female will only release one egg at a time. So when you're inducing a superovulation, it is going to release many eggs at a time, and after mating with a male, you're going to get many embryos. These embryos are collected and then microinjected with the DNA of interest. Then the embryos are transferred into recipient females using different methods. Then in this yield, what is going to happen is 1 to 2 transgenics from the 100 micro-injected and transferred embryo is going to be there, because see, you never know the sustainability of the embryo in the female, if it is going to be there or not, the viability or the survivability of that embryo to be formed into the progeny. So lower in mammalian species and very low in ruminants. Ruminants are your cow, buffalo. These are the ruminants, ruminating animals. They have 4 stomachs: rumen, reticulum, omesum, abomesum. So the process of superovulation, mating, isolation, one cell state zygote, microinjection, oviduct transfer, and identification of founders. So that is, you will identify whether this is there or not using a PCR or a southern blotting technique. So in other mammalian, non-mammalian species, the pronuclei are not exactly visible. So DNA is going to actually be injected into the cytoplasm of 1-day-old embryos. Then this is going to see that whether there is of course, when you attach a promoter to it, you will be able to see whether that particular gene has been expressed or not. Now, for the number of eggs to be produced of a superovulation, what are you going to do is, you're going to take a serum on the follicular stimulating hormone on first day, and the human chorionic gonadotropin or the luteinizing hormone on day 3, and then this is mating with the fertile male on day 3. Because of which, whatever fertilized oocytes are there, they're going to be micro-injected into the day 4 with foreign DNA construct. Then the micro-injected eggs are going to be implanted on the same day, that is on the day 4 itself, or they can be incubated or overnight, and then implantation into the oviduct of the pseudo-pregnant female will take place. See, we have to induce the pregnancy conditions into the particular mammalian species, because usually what will happen is, at every point of time its mating cycle is not there, especially in the animals. So because of which, you have to induce those conditions and then only implant it, so that the animal also, because of the presence of or given all these hormones, will start thinking that it is pregnant. So because of which, what will happen is, it is able to have or sustain that kind of atmosphere within the oviduct or within the uterus of the mammal, because of which there is a chance for the pregnancy to be to come to what do you call it, till the end, or production of progeny is there. Superovulated female mated, female male pronucleus is there. This is how you're going to hold with the fertilized egg with the pipette, and then you're injecting the gene, transgene, into the pronucleus. So usually this viability is not going to be seen, but only the chance of around 1 to 30% can produce a live transgenic animal that is there. So it is very less that you get to see. So when are you going to make or generate a pseudo-pregnant female? That is, female mice are going to be tricked, so that they think that they are having they are pregnant in this first place. They are put into that estrus condition and then mated with a vasectomized male. That is because we only want this particular gene to be expressed. So since we have already taken the zygote or the embryo over here, we are going to let it be mated with a vasectomized male, so that its sperm does not reach, so that finally the organism or the rat is the mice is actually thought that made to think that it is pregnant. Then When we implant these particular embryos into the mouse, then of course it will become truly pregnant and will try to give birth to the live offspring, again depending upon the viability and the conducible conditions of the embryo. And then this is how you're going to get the embryo, 200 to 250 cells in fact. And then you get to see micro pipetting of these cells that are taken, basically embryo cells. And then you can introduce the transgenic constructs into this by electroporation or different kind of viral vectors. And then culture the cells in presence of an antibiotic so that other transgenic the transgenic uh, cells are going to be retained while the others are going to be killed, and then insert it into the blastocyst of the mouse. And if at all you want an albino, then you're going to take it from the original stem cells of the pigmented mouse and try to inject. So you have donor blastocysts, you're going to culture them, and inner cell mass is taken over here. So finally, your embryonic stem cells are there. You're going to see that through electroporation or viral vectors or through transfection, you're going to insert them into your embryonic stem cells. With this, you're going to get a transfected cell. So again, blastocyst, holding pipette, embryonic stem cells, and then you're going to inject them. This is how it is. This is the actual picture in when you're taking an albino along with a normal gene, that albino gene plus your normal gene, you're going to get a chimeric mouse having both albino, which is being expressed, so you do not have any skin pigment, and your normal pigment, because of which you're going to have a pigmented section along with it. So it's this kind of a mouse is known as a chimeric gene or a chimeric mouse. Then when you start breeding offspring of the chimeras, their offspring will have all the three types of genes, genotypes, meaning wild type homozygotes, heterozygotes, and transgenic homozygotes. So they are going to be completely pigmented. So this is how you're going to have. This is a female wild type black mouse. This is a male chimeric mouse. Finally, in the first progeny, 3:1, you're going to get a completely homozygous recessive, which is going to be black, and 3 heterozygous or brown mice. And then these heterozygous, when they are mated together, again you will find that they have a mating, like you're going to get again 3:1 progeny. So you get a picture of this black mouse with no embryonic cell integration, and chimeric mouse, you can just see the black patches in the skin. And then this is a chimeric mouse having a low embryonic stem cell integration. So this is a complete black, and this is not exactly, but it leads to a chimeric in nature. Then we have the second method wherein we have taken the embryonic stem cells, then selection for desired, expression of desired gene, and then injected into the blastocyst, implantation into the uterus, and then you have taken the method that is, pronuclei with designed gene vector implantation, testing for offspring. And then the third method is your DNA transfer via cloning. So what is happening? Foreign gene taken, transferred into the somatic cells. That is, you have two types of cells in the body, which is your somatic cells and germ cells. Your germ cells are from the reproductive organs. Your egg cells and sperm cells are your germ cells, whereas all the other body parts having cells are your somatic cells. So whenever the foreign gene is introduced into a somatic cell, and then the nucleus is introduced into the cytoplasm of an enucleated oocyte, from the somatic cell into this, you'll get a transgenic clone. Basically, you're again looking at the how Dolly was created. So in 96-97, Dolly was created. It is the first living offspring created from the differentiated cell, in fact. Now we have the fifth method, which is the sperm-mediated gene transfer, wherein sperm cells are having the capacity to bind to just basic naked DNA, or that DNA can be bound to liposomes, which is a vesicle. So in 2002 and in 1989, two papers were released regarding this. And these sperm cells then are used for introducing exogenous DNA into the oocytes through in vitro fertilization. So finally, this is the sperm cell mediated gene transfer. Retroviral technique is wherein you have the dsDNA incorporated into the host. Similar with lentiviral, basically say like low expression, wherein your gene locus wherever it is there, it is going to be silenced. So finally, you're going to get stable transgenic lines with using these two. And then we have one more technique, which is your electroporation technique. So you can expose the cells to pulsified electric field. In fact, just give pulse of electric field pulses. Then because of which there is a potential that there's a polarization occurring. Therefore, what will happen is permeability of the exogenous gene to take place. It to go into the particular cell is going to. So when you combine other methods along with this particular electroporation, there's been a high success of the transfer to take place. You have one more chemical technique, which is calcium phosphate. So you're going to use that, or DAE, that is your diamino ethyl dextran, that is going to be used, and transfection, meaning in the chemical itself, you have the DNA in it. And then with the help of this chemical, you're going to see that this DNA or the gene fragment is taken up by the, the host cell, that is your, the cells. Now you have something called as RNA interfering genes or small interference RNAs. siRNAs are as they are known as. They're only around having 20 to 25 nucleotides long, and they bind to the complementary sequences on the mRNAs. When they are bound to the mRNAs, what do? They stop the expression of the mRNA gene, because of which you do not have protein translation. So what is happening? Because there is no mRNA translation into proteins, you do not have production of proteins. So this can be used, small interfering RNAs can be used for either seeing that there is a stable gene expression or for knocking out of any kind of target genes. Now, when we look at transgenesis, when we are looking at all these, we see that, it still has quite a lot of cons, in fact. So if at all, when you insert a gene, there can be some kind of insertional mutation, which results in alteration of the different kinds of processes. And when there is unregulated gene expression also, it results in improper expression of the gene products. And of course, in the transgenic animals also, they have a lot of side effects like arthritis. They have dermatitis. The skin has become problem, like they are having skin issues, hair loss, and cancer, and heart problems, in fact, in the transgenic animals itself. And whenever there is integration of DNA into the Y chromosome, what will happen is, since it's only in the Y chromosome, only the males will get it. So this is also not going to be so useful. Now, when we look at the applications of transgenic animals, we have something called as the transformers. Transgenic animals engineered to produce the therapeutics such as insulin in the milk, vitamin A enhanced milk. So proteins in the mammary glands are going to be increased in nature, and then finally through the milk harvested. So around 1 to 10 grams of protein per liter of milk could be yielded. So Hermann the bull was genetically engineered to carry the human gene for the production of lactoferrin. Because of this, it is very much important for the immune system in the infants, as it is present in the mother's milk, but it does not occur naturally in the cow's milk. So this is Hermann the cow. And then we have transformer goats, wherein, they could have or they could carry the recombinant human antithrombin 3. So it's a blood thinning low protein. Around 5 grams per liter of antithrombin 3 protein was produced in the milk, and it got marketed as atriene. So by a company called Genzyme Transgenetic Corporation. So it became the world's first FDA approved drug in the year 2009, in fact. So these were the three goats that were able to produce this particular protein. So it's a blood thinning protein. Now you have transformer sheep also, which produced a human alpha 1 antitrypsin, which was a medicine which is used against a potential medicine. So we never know whether it is actually going to be functional or not against one particular treatment of a disease called as cystic fibrosis. So, cystic fibrosis is a disease which actually causes inflammation and thickening of the lung tissue and the mucous membrane surrounding the lungs, basically. Because of which, what will happen is you are not able to breathe. Production of salt is there, and then thickening of the mucus is also taking place. So this alpha 1 antitrypsin is going to inhibit a lot of proteases, that is, those which break proteins, protects the tissues from different kinds of inflammatory cell enzymes, and especially it acts on neutrophil elastase, whose concentration will actually rise whenever there is any kind of acute inflammation in the body. In its absence, that is, in the absence of alpha 1 trypsin, the neutrophil elastase is going to break down the elastin, contributes to the elasticity of the lungs, because of which what is going to happen is you will have something known as the COPD, that is chronic obstructive pulmonary disease, which can be seen in adults, and also cirrhosis. Cirrhosis is a disease wherein you know hardening of the tissue, which takes place in either adults or children also. So this is the sheep, and then which could produce alpha 1 antitrypsin, in fact. And then you have the different kinds of proteins produced from different kinds of sources and which are used against different kinds of diseases. So I should say that it is a beautiful fact that we are able to extract or such kind of substances just by consuming either its products, so that there is some kind of a wound healing, in fact, instead of just taking your conventional medicines. We also have a pig that produces sugar alpha 1 galactose that the human body is going to think that it is a foreign, and so that it rejects the organ. So what is going to happen is this is going to knock out the genes that encode this glycosyl transferase enzymes. So what happens is sugar is added onto the organ surfaces, so that it is not going to be accepted, or it is not going to be identified as a foreign entity and will be able to save. So it is a method to bypass your organ rejection. So these were the pigs that were produced. Of course, food sources, superfish, having salmons were produced. You can create all the animals that you want so that they can grow quick for human consumption with more nutrients in it, so that if you just have that, your daily dose of vitamins, minerals, and your carbohydrates, proteins are just going at one go with having only one particular amount of food rather than all. Example is superfish with growth hormone, and see, you can just see the genetically altered size of the fish where as compared to the normal salmon. Of course, you have a lot of scientific biological methods that are used. You have a super mouse, which is bigger than that of the conventional mouse. You have a smart mouse which can think properly based on as per when compared to the conventional mouse. And then you have some incorporation of a glutamate receptor and gene NR2B that is going to help in the learning and memory easier. So ability to recognize objects, learn more effectively, spatial learning, all this can be done. And then you have glowfish. Fluorescent fish, zebra fishes, they were the first glowfish. So you have different colors and they look really good. Most genetically modified animals that are publicly available right now, all such kind of beautiful fishes. And then they're used as disease models. So you have Huntington's disease, which is a neurodegenerative disease, autosomally inherited, in fact. So because of that, you are for sure that this particular disease is going to be transferred, because it is not allosomally transferred. It is for sure going to be transferred to the project. So because of this, there is a mutation in the Huntington gene. So you have a CAG triplet nucleotide duplication, which causes a buildup of the polyglutamine amino acids in the neurons. So when you have this, you can induce that in the transgenic mice, and then you can construct genes having the Huntington's disease, and then start giving it medicine, so that experimentation can be done. Of course, you can induce Alzheimer's, AIDS, Parkinson's also, so that accordingly you can just see. And what about poliovirus? Because humans, you have the receptor, but in the mouse, this particular viral receptor is not there. So transgenic mice having the human gene for the receptor can be infected by poliovirus, and this will actually cause paralysis in the mouse. So that is a mouse model. So these are the different mice models that can be used to find out other basic transgenic animals. In fact, we will just have a look at it also. I'm supposed to start with stem cells. Today I will be taking 10 minutes more of the class, so please bear with me. We have seen the different kinds of applications, so this I will just go through quickly because it is just going to be a recapitulation. You can see preparation of the transgenic animal, definition of it has been given, the history, and I'm sorry, it is the same slide I'm just showing. Give me 1 minute, please. So basic growth hormone inserted, and then you have fluorescence gene inserted, so that you can see wherever it has been inserted. Methods are there: DNA injection, transposons, viral vectors, and ICSI, and then use of episomal vectors and pluripotent cells. All of it can be used. So ICSI is known as your intracytoplasmic sperm injection, in fact. So like I told you, glowfish, glow rabbit, and different transgenic animals having a mouse with the ear also has been grown. Transgenic monkeys have also been seen. So different kinds of, in agriculture, in medicine for xenotransplantation, nutritional supplements, pharmaceuticals, human gene therapy, agriculture for improving the breeding and the quality of the animals, and to incorporate disease resistance. And then you have the toxicity sensitive transgenic animals. So goat with a gene of spider silk, in fact, advantages you can just see, and the disadvantages also. So summarization is that it is necessary for the human welfare. Of course, ethics committee will think about it in a different manner. So this is about transgenics. When it comes to stem cells, an overview of the stem cells, when you get to see that stem cells are an undifferentiated mass of cells that form various cells of either the same or a different kind. Tausto? The simple or complex organ cells are able to be differentiated into different cells, and they are capable of forming a complete entire organism. As such, they are able to produce different cells, repair, replace damaged tissues. So potent cells capable of differentiating into other cells, and then a single cell can be turned into different types or specialized. So usually we have around 37 trillion cells in our body, and out of that only around not 2%, that is around 744 million cells happen to be stem cells in the adults. So blank cells is what they are known as because they can follow any kind of path to form either lymphocytes or like what do you call it, other types of cells. So you have something which is known as the Hayflick's limit, wherein it shows about the division in a finite amount of time. So these stem cells do not follow that particular Hayflick's limit. And then you see that, different lineages are there. You have lymphoid precursors, erythroid precursors. So a stem cell can become or undergo some phenomenon which is known as transdifferentiation, where it can either form a lymphoid or an erythroid precursor. History started with around 1908 itself. First animal created using IVF technique was around in the year 1958. Then the finding of stem cells also took place, discovery of hematopoietic stem cells, and whether bone marrow could be transplanted between two siblings with severe immunocompromised deficiency, that is your SID disease, and then the concept of tumor cells. Of course, then we came to know that in the year 1978 itself that our human umbilical cord blood were all hematopoietic stem cells. And then no Nobel Prize was given in the year 1981. And in 1992, you have different stem cells that were identified in the human brain also. Of course, in the 2001, what did they do? Embryonic stem cells of the mice were created using the or from this nuclear transfer technique. And you can just see 2006 discovery of induced pluripotent stem cells, and 2008 first human embryonic stem cells without any kind of the embryo to be damaged was discovered, isolation of the cells. Then say 2017 you could see that stem cells could be used to see that you could start hearing again and make the human blood-brain barrier, which is one of the most important structures or entities that are there in the human brain, so that they are protected against different kinds of infections. Very few substances actually are able to cross the human blood-brain barrier. When we come to the classification based on the potency, so based on its function, you can classify it as totipotent, pluripotent, multipotent, oligo, and unipotent. Basis of source, embryonic, adult, and induced pluripotent. So a totipotent stem cell is able to differentiate into any organ or tissue. So it's something similar to zygote and is present in the blastula stage in the gastrulation. Pluripotent stem cells will give rise to cells in the body, and they are they are going to be as seen in the kind of in vivo cell, fertilized egg. And the 8-stepped, this is your basic totipotent. But when you obtain it from the blastocyst, then it is going to be known as the pluripotent cells, which can form neural cells or cardiac muscles or blood cells. And then induced pluripotent cells, they are going to be prepared artificially. So you're going to take an adult fibroblast cell and then try to reprogram it. Then you're going to get induced pluripotent cells, which can be forming your myocytes, adipocytes, neurons, neural cells, pancreatic beta cells, which produce insulin, motor neurons, and hematopoietic progenitor cells. So these are the different functions. So example, these multipotent cells will be forming all your erythropoietic stem cells, such kind, so that they are present, which are going to form your blood, fat, and different kinds of muscles. When it comes to unipotent stem cells, they form single type of cells. Example is a gamete. This forms into cells in the male. So from this hematopoietic stem cell, you're going to get only platelets after differentiation because of the different kinds of interferons, interleukins that act. And depending upon that, you're going to get only one type of cell from this. So this cell lineage, whatever is coming, is because of a different kind of interleukin or interferon that is going to act. Embryonic stem cells, they are going to be there from the embryos that are going to be derived, and then that have been fertilized in vitro. And so that, from this, you're going to get different kinds of cells altogether. So these are the undifferentiated stem cells. Adult stem cells are also undifferentiated cells found only in certain amount of tissues that are present in fact, so that what will happen is they only have the regeneration capacity. So they are also known as your somatic stem cells because they are not germ cells, as they are not sperms or egg, and they are present only to heal or regenerate themselves. So now what is the use of stem cells? You see that it is able to damage the different kind of whenever there is a damage in the cells, that cells can be regenerated. So whenever cancer comes and is what do you call it, you have a cancer therapy, it will stop the ability to produce new cells is stopped. So when that case scenario, if you use stem cells, the new cells of the host can be developed. Then so that's and then they are either they can be used as disease models, and then of course to study the different kinds of diseases. So somatic cells from the patient, then you're going to make an induced pluripotent cells. You get the different kinds of progenitor cells that are there. Then you get different kinds of neurons that are there, and then you can either use it for disease modeling, drug discovery, or cell therapy. This is one thing. So types of stem cells we have just seen. I'll just like show you the different kinds of PowerPoint that has been there, so that you'll just have you can just see the induced pluripotency. Patient from the germ cells. Oocyte, fertilized egg, morula, totipotent cell, blastula, embryonic stem cell, induced pluripotent stem cell. Somatic cells are going to form these. So these can be differentiated and either be used for transplantation to after a new organ has been produced. When it comes to amniotic stem cells, there is something called as the amniotic stem cells. So in this, I do not have a better picture, unfortunately. You will get cell proliferation. This is your P53 suppressor protein. Let me just see if at all I can do something to this. So you have a blastocyst, embryonic stem cell, then the endosperm, mesoderm, and endoderm, all giving rise to the different cells. So like I told you, you can just culture them into endoderm, mesoderm, as well as your ectoderm. So that can be used for basic research or drug development or cell therapy. So types again, like I told you, embryonic stem cells, induced pluripotent, and adult stem cells. So the basic function, how it is going to be characterized is being given over here, induced pluripotent cells, and your iPSC, that is how is induced pluripotent stem cells being created. And then what are mesenchymal stem cells? That is from the bone marrow stroma. You're going to take a multipotent stem cell that can be differentiated into only bone-related products. That is either your osteoblasts, chondrocytes, or adipocytes. And then these are used in clinical, applications. That is either hormone or grafting or immunosuppression reaction. And potential therapy is basically graft versus host disease or Crohn's disease, which is an autoimmune disease, if I'm not wrong, bone defects or different kinds of muscle. Because you're also getting a myoblast over there, you can use it for cardiac repair or tracheal repair. So hematopoietic stem cells will give both lymphoid and myeloid precursor cells. So anything can form with the hematopoietic. Whereas if a neural, only neurons, astrocytes and oligocytes are going to be formed. Cancer stem cells to induce cancer, and then in case that is going to take place, you can use it for treatment of cancer. So only the chemotherapy resistant stem cells are going to be killed, and so that tumor will also be resistant to the chemotherapy. So then what will happen is, in these kind of cells were actually identified in the tumors, that is brain, breast, colon, ovary, all these, whatever tumor was there in those, this cancer stem cells were found. So you could surgically remove it. So it says that if at all this treatment of cancer is not going to remove this cancer stem cells, then the tumor will reappear and the chances are a lot. So this is the summarization of stem cells, and they have applications in research, drug discovery, and cell therapy also. So this is all about your different kinds of stem cells. In fact, now I'll be talking about the last topic for us, that is your gene therapy. So where exactly is gene therapy going to be there? Just give me some 10 minutes. Let me finish this particular topic. There are around 20 or so slides, which can be done quickly. So experimental technique gene therapy is for what? To see that, if at all there are any defective genes in the body, that can be tried to, repaired or corrected. So most common is insertion of a normal gene in place of an abnormal gene. Other approaches is, you are, whatever, if at all there is a mutated gene that is going to cause a disease, you replace it with a healthy copy of the gene. And then you inactivate or knock out a mutated gene that is already there and which is not functioning properly. Then you introduce a new gene into the body to help fight the disease. So again, what is it, or where are they studying this gene therapy? For severe combined immunodeficiency, wherein you have a lot of quite a number of genes that have been knocked out. Hemophilia, that is a blood disorder. Parkinson's disease, that is again your neurological, I shouldn't say neurological, it's a protein folding disease, wherein you have misfolding of protein. Of course, cancer, either increase of the oncogenes or suppression of the tumor suppressor genes. And then HIV. All this people are studying for gene therapy. History is, again, we should say that 60s itself concept of gene therapy, and people tried to do gene therapy from 1970s itself, but finally in the year 1990 you could see that people tried to use genetic gene therapy, so that immune system deficiency in a girl called Ashanti De Silva happened. Now you have this to treat thalassemia, cystic fibrosis, certain cancers, and also sickle cell. 90s, 96, 2007 to 2011, you have around, retinal disease or color blindness, adrenoleukodystrophy, all of this being treated. 2011 also, there could be a cure for HIV using gene therapy. That is what they try to find out. So let us see the different types of gene therapy. You have germline gene therapy and somatic cell gene therapy. In germline gene therapy, you're going to have a permanent change potential for this particular permanent gene, so that whoever has this gene which has been inserted can be passed on to the progeny. So this has certain ethical issues about it. So what is going to happen? So the sperm producing cells are removed from the defective gene that contain the defective gene in a man. Then healthy gene was tried to replace the defective one, and then put them back into the mouse's testes, where they could get matured inside the mouse, and then they could start producing the healthy human sperm, which again after testing to see, and they were used to in vitro fertilization using eggs from the woman, and then resulting embryo was implanted, and the child, the progeny was having birth, was having genes from the father, but without the father's disease. When it comes to your somatic gene therapy, only the targeted cells in the patient are being cured. So what is happening? It is the somatic cells, but not the germ cells. See, I will have to clearly tell you again, because somatic cells are your all the other rest of the cells that are there in the body. Your germ cells are the egg cells and the sperm cells. So these somatic cells, when they are having any kind of gene therapy, it is only in the body. So it is not being passed to the progeny. So transportation of the gene to different target cells is an issue, and whether any kind of side effects are there, all of this has to be taken into consideration. Again, when it comes to somatic gene therapy, you have two types: ex vivo and in vivo, and they're modified outside the body, transplanted again, that is your ex vivo. And because the cells are treated outside the body only, it's got the name that it is ex vivo. In vivo is genes are changed in the cells within the body cells itself. So you can see cells are removed from the patient. Then in the lab, the virus is going to be altered so that it cannot be reproduced. Gene is inserted into the viral cells. Then viral cell is used to mix with other cells from the patient, or from the the person, in fact. Then genetically altered and altered cells are injected that produce the desired protein or hormone which has not been there earlier. So ex vivo is basically you're going to use a DNA and viral targeted cell. Non-viral also you will have. Direct is in vivo, wherein you take the viral DNA, insert it into the human being. Non-viral also in the same manner. So basic process of gene therapy is using viral vectors. So you can use the DNA delivery into the cells for gene therapy, and it can be using viral vectors and non-viral vectors. So this virus is something which is going to infect. So that is the reason why scientists have tried to use this particular ability of the viral cells to remove the disease causing genes, and then you can insert the therapeutic gene into it. Then viruses bind to the host. Then this genetic material is definitely going into the host cell. Then by removing the viral DNA, you can use the virus as a vehicle or a vector to deliver this therapeutic DNA into the host genome. Then viruses are used or altered to make it safe, in fact. So of course, certain only a little amount of risk is there. So you can use adeno-associated viruses or retroviruses, pox viruses, alpha and herpes viruses, all to deliver your desired gene. Now, what are the different types? Again, this can be either temporary or permanentary. Different types have been given over here. You can just see adenovirus, adeno-associated virus, the type of DNA that is included in this, and what is what the characteristics of the viral is also given. And then what is the family host genome interaction and the packing capability. Then this is the cells with original genes. Vector inserts new gene into the cell, new gene in the cell along with original genes that is present. So it's a very common tool, can be performed either in vivo or in vitro, in fact. And this viral delivery is known as transduction, and the cells that are infected with the gene are called as transduced cells. This is how the viral vector binds to the cell membrane, packaged inside the vesicle that is there, then breaks down, releasing the vector, and this is going to go into the nucleus and alters the cell. Non-viral vector, you have a physical or a carrier free gene delivery and chemical approaches that are present. And then you have chemical approaches, which is synthetic vector-based genomic DNA. Of course, again, you have physical approaches, which are your electroporation, similar to your gene transfer methods, in fact. The needle injection, gene gun, ultrasound, and hydrodynamic delivery. All employ physical force so that, the cell membrane is going to be permeated and your gene of interest is going to be transferred. So simplest method is take a naked DNA and and then inject it into the cell basically. So transfection. So you can use gene gun method or electroporation, and then DNA coated gold particles into the cell using high pressure gas. Chemical methods, lipoplexes and polyplexes. Lipopolysaccharides are used basically. Again, and then you have lipoplexes, wherein you can take in the plasmid DNA, cover it with the lipids, in fact, or formation of micelles is taking place. Then you have anionic, neutral, and cationic lipoplexes. Then these are used as synthetic vectors. Little toxicity is there because it is a vector, it's a missile basically. And then, you can use it. Cationic vectors are basically having a positive charge, and they are going to have form a complex with a negatively charged DNA also. They interact with the cell membrane, because of which the lipoplex endocytosis is going to take place, because of which the cell will release, this lipoplex is going to release the DNA into the cytoplasm. Then the cationic lipids are also protect against the degradation. So what is the commonly used lipoplexes? You can just see for gene transfer. You use your in the it is also going to help in decreasing the activity of your oncogenes. It is also helpful in the respiratory epithelial cells to be transfected, so that your genetic disorders such as cystic fibrosis will be cured. So with DNA, whenever they are present, they are known as polyplexes. They consist of cationic polymers and they are regulated by ionic interactions. So you have co-transfection with the endosome, lytic agents that are occurring in the polyplexes. So you could actually, with the help of gene therapy, treat blindness and Parkinson's disease. So whenever this happened, what they did was, there's an abnormality in RPE65, congenital, that is by birth itself. In the first few months of life, there is progressive loss of vision. So you take that virus, which is harmless, inject this particular functional gene into it, and then completely, transfer it. Now, in the case of Parkinson's, even today, you have certain drugs that were introduced just a couple of weeks or 1 week ago, in fact, for Parkinson's disease and for Alzheimer's also. So what happens is, over here, there's a weakness in the motor skills. So this is going to be they try to produce a GABA, that is gamma aminobutyrate, which is an inhibitory chemical that could actually stop this brain and reduce the tremor. How is it going to work? You're going to give a inactive virus AAV2 along with the subthalamic nucleus into the patient, in fact. Then this gamma aminobutyric acid neurotransmitters are going to be made so that normal chemical balance is maintained, because of which the disease progress will be at least, reduced. Of course, whenever there is any genetic disease, normal life can be given to such kind of baby with that kind of hope that we have. Then healthy life to a cancer patient, of course, any other kind of diseases. But we have certain disadvantages also. What are they? Increased rate of abortion, cost is very high, insurance should be able to cover it, and then of course it is there are so many other things which are there. And then you have ethical questions also. Is it good or bad that we are doing gene therapy? In fact, is it normal, or will it actually contribute to some other disorder, in fact, that we do not know of? And is it going to be any kind of why should we normalize people who are having why should we actually stigmatize people who are having any kind of genetic diseases? And will they start using gene therapy to actually enhance beauty, intelligence, or athletic ability? All this is, these are the ethical questions surrounding the gene therapy. Now, this is all I have regarding the gene therapy also. If you would like to have these particular slides, then you please put in a message in the WhatsApp group pertaining to your class or course. In fact, then I will be able to share these in case you're interested. So that's all for today, and that's all. I have completed my animal biotechnology and immunology topics altogether.
Excuse me, ma'am.
Yes.
Ma'am, what are the two methods of gene therapy, ma'am? What are the methods, ma'am?
I have just shown it to you in the PowerPoint. In fact, if you get to see gene therapy, two methods are in vivo and ex vivo. In the type of
Okay,
in
ma'am.
that is there in the types of gene therapy include germline gene therapy and somatic gene therapy. And when it comes to somatic gene therapy, you have ex vivo and in vivo.
Ma'am, actually, you have missed fetal stem cell and adult stem cell, ma'am.
The adult stem cells, I have told that also. In fact, overview of stem cells. I said about embryonic stem cells or the fetal stem cells
Yeah,
that are going to be there, and the adult stem cells also. So all the topics in your textbook have been covered.
okay, ma'am.
All right. Yeah. Thank you
Okay,
so
ma'am.
much.
The PPT slides will
Thank
be
you,
better
ma'am.
for us, ma'am.
Yeah, if you please can message in the official group that has been created, then I will send them to you. All right.
Yeah, it's already been created. If I got your phone number, then I can just add you on the group so that it will be easy for us.
All right. I'll just put it in the chat box so you can just see what is.
Sure, ma'am. That will be good for us.
I think I just put in my phone number.
Yeah, yes, ma'am. You got it. Thanks a lot.
All right. Yeah. Thank you so much.